All right, well, we're now into the second half of the semester. So I hope you all enjoyed the midterm and it was everything that you had hoped it would be. We are grading those and we'll have those for you by Thursday is the goal. And, you know, we have switched gears now, as I said, talking now about population genetics. In the last two lectures, you've watched videos. The first lecture being on Hardy-Weinberg equilibrium, which for many of you was likely a review. And then our last lecture from last week was on inbreeding and the consequences of non-random matings in human populations. And so hopefully you were able to watch that video, and that's going to be important material for the assignment that we're doing this week. We're going to continue now talking about population genetics today. And let me share my screen here. And so I'm going to talk about two topics today. So really what we're interested in, the question of in today is what controls the fate of a new allele that is introduced into a population. There are two primary forces that we want to talk about today. And the first of these is random genetic drift. So who's ever heard of genetic drift? So it's a really important concept in population genetics, but it's one that you're well equipped to understand today. Because really what it is just changes in allele frequencies between generations that's just due to the process of sampling. So when we talk about sampling from the gene pool in population genetics, that means that we are randomly taking alleles or genotypes from the gene pool in order to make the next generation. And so the first concept that I'm going to talk about today is the concept of random genetic drift as one of the primary forces that determines changes in allele frequencies between generations. So to do that, let me try and introduce that concept. All right, so in POB, you already, and in anthropology, okay, you've encountered genetic drift, so that's good to hear, and AP Bio. So for some of you, this might be a bit of a review, but you know what we're going to learn today is the foundation for being able to actually simulate genetic drift in the computer, which is really fun. Okay, so let's think about the just an intuitive introduction to genetic drift. And so one way to make things intuitive is always just to think about yourself, right? So let's think about your parents. Think about your parents and your parents, the two people that comprise your parents. Let's think about them as a population. And that's our population. And let's consider a single locus, A, and both of your parents are heterozygotes. So they are big A, little a, okay? So dad is big A, little a, mom is big A, little a, and therefore, when we compute the allele frequencies, we say P equals Q equals 0.5 in this case, okay? Because we have two big A alleles in our population and two little a alleles. All right, so now let's imagine the formation of the next generation, which comprises yourself, so that's one person, and your sister. So you have two people in the next generation, and unfortunately, and this is often the case in theoretical population genetics, your parents are deceased, okay? So there's only you and your sister left in the world. All right, so let's think about what are your possible genotypes. Your possible genotypes, given that your dad was a heterozygous and your mom was a heterozygote at the A locus. So your possible genotypes, you could either be big A, you could either be big A, little a, or you could be little a, okay? And similarly, your sister's possible genotypes. Again, could be big A, big A, big A, little a, or little a, little a, okay? And so we can think about different scenarios depending on the genotypes of you and your sister, given that your parents were heterozygotes, and now you and your sister have been born, your parents are deceased. And let's think that in the scenario that, so if you are a heterozygote and your sister is a heterozygote, then, and our population just has you and your sister, and then we have 2 copies of big A and 2 copies of little a, then the allele frequencies have stayed the same. Similarly, if you are homozygous for the big A allele and your sister is homozygous for the little a allele, then again, the allele frequencies have stayed the same in the next generation, or the vice versa. However, if you are big A, and your sister is big A, then the allele frequency has changed in one generation. Or similarly, if you are little a, and your sister is little a, then again, the allele frequency has changed in that one generation, okay? And this isn't because of anything magical. This is just sampling from the gene pool. We have sampled from the gene pool that was comprised of your parents. We've randomly sampled from that gene pool, and we've generated you and your sister, and you have a given genotype at the A locus. And depending on which genotypes you end up having, we either have the allele frequencies remaining the same, or the allele frequencies are changing between that parental generation and your generation, okay? And this is just because of random sampling. And so this is what random genetic drift is just changes in allele frequencies due to random sampling. Let me write that again. And it's a random genetic drift is just changes in allele frequencies between generations due to sampling, the process of sampling. All right, so let's imagine an even simpler case, and we can do this as theoretical population genetics. The beauty of population genetics is that we can do Gedanken experiments, that is, we just think about things and think about what the likely outcome would be, as we did with Hardy-Weinberg equilibrium. And so we can imagine scenarios that, you know, aren't necessarily plausible, biologically plausible, but they are informative. And so we can imagine, let's imagine the simplest possible scenario, which is imagine a population, a human population of one individual. And that individual is a heterozygote, big A, little a. Okay, and so let's define that population. So in this population, we have 2 alleles. The allele frequencies are equal to each other, and we use P and Q to define the allele frequencies. And the population size is 1. Okay, and this is our initial generation, which we'll refer to as T0, the first generation. And so a question we can ask then is, given that we have this population of 1 individual, and we're in the theoretical realm here, and so we can generate a new population just from a new generation from that one population, we can ask the question then, what is the probability that population will drift through this process of random sampling to P equals 1 and Q equals 0 at T1? What we're asking here, when P equals 1, the allele frequency equals 1, this we give a special term to. We call this allele frequency fixed in the population. And that is the scenario in which the allele frequency has gone to 1 in the population, and then there is no longer any genetic diversity, any additional diversity at that locus, okay? And so when we do this, we have to assume, again, that population size remains at n equals 1, okay? And so this is typically the case in many population genetic simulations and theoretical considerations. We just consider the population stays the same across time, and that each generation, once the new generation is formed, the previous generation, they're all dead and they're all gone. Just like in the tragic case of you and your sister and your parents, okay? And so this is the scenario here. Our population size stays the same, but we can form new generations. And so let's assume in this case, or let's consider, like, what is the probability that the population allele frequency is going to drift through the process of random sampling to fixation to an allele frequency of 1? Well, to make our one individual in our next generation, we have to randomly sample 2 gametes from our current population. And so we just reach into the gene pool and randomly take a single gamete, and the probability that it's going to be have the big A allele is 0.5, right? Or P. And then we need to make a diploid individual. So we need to reach back in and randomly sample again. And when we randomly sample again, the probability that we have the big A allele again is P, the allele frequency. And so it's 0.5 squared, which is equal to 0.25. So in our theoretical scenario, the probability that P is fixed goes to frequency 1 in one generation is equal to 0.25, or 25% of the time. And conversely, in this simple scenario, the probability that Q is fixed in one generation is also going to be 0.25, okay, in this simple scenario. Let's make it more complex now. Let's consider something more realistic. Let's say, what if our population is actually 2 individuals with P equals Q allele frequencies, so they're equal to 0.5, and we can ask this again. What's the probability in one generation that the allele frequency of P is going to go to 1? That is the allele will be fixed in the population. So again, we think about randomly sampling from our gene pool, randomly taking an allele to make a gamete, and with probability P, we're going to take one of the big A alleles. We have to make 2 individuals, and so now we multiply by probability P to get our first individual. These are independent probabilities. We're going to make another individual, so we have to randomly sample again, and with probability P, we will get the big A allele again. And then to make another, here's our 2 diploids. So there's our 2n. I won't use n because that's what I'm using for my population size here. This is individual 1 and individual 2. And so to compute the probability that I just sample the big A allele from our existing generation to make our new generation of 2 individuals, it's going to be 0.5, our allele frequency, the power of 4, that's 0.0625, okay? And so that tells us that the probability, given that we have 2 individuals and allele frequencies of 0.5, the probability that that allele, that A allele, will go to fixation in one generation is equal to 0.0625 in this case. And this generalizes. So for example, what if we had n equals 2 with, in this case, P is equal to 0.9, if our allele frequency was 0.9. Again, it's the same scenario that we have to generate 2 diploid individuals. Each time we're drawing from our population. In this case, we make the same calculation, but in this case, it's 0.9 to the power of 4. So that is equal to 0.6561. And so this simple scenario then generalizes that given some allele frequency P to the power of, because we have to make diploid individuals, multiplied by the number of individuals, this is equal to the probability of the allele going to fixation in one generation. And so with this simple formula, then we can ask, what if we had p equals 0.5, like in our first population, but now n equals 10,000. So we have a more realistic human population size. In that case, the probability is p to the power of p, so 0.5 to the power of 2 times 10,000, which equals 0.5 to what, 20,000, the power of 20 to the power of 20,000, which is a very, very small number. I can't compute that number for you. Very, very small probability. But what this illustrates is that drift, this random sampling process that's going to change the allele frequency, differs as a function of the size of the population. And drift, genetic drift, is weaker in large populations. So we can talk about the impact of random sampling or genetic drift on changing the allele frequencies. So in the case of having a population size of 1, we can see that it's reasonably likely that we're going to go to fixation in just a single generation, whereas if we had the same allele frequency of 0.5, but we have a much larger population size, the probability that it's going to go to fixation in a single generation is much, much smaller. Let's then ask a different sort of question, okay? Let's consider now a different scenario. Let's consider a population where the population size is 500 individuals, but again, P equals Q equals 0.5 in our current population, T0. So this I'll use to refer to the current population, or sometimes we could also say Gen 0. Okay, so in this population then, what does this mean? This means, therefore, in this population, we have 500 copies of the big A allele, and we have 500 copies of the little a allele. And so we can ask a different question, not now about going to fixation. We can ask the question about staying the same. And we can ask, what's the probability that the frequency of A stays the same in the next generation? What's the probability that the frequency of the big A allele stays the same in our next generation, which we'll call T1? Given in our theoretical population that the population size stays the same at 500 individuals, 500 diploid individuals. So for P to remain at 0.5 in T1, how many copies of the big A allele will we need? That's a question for you. So we need 500, and how many total alleles are we going to have in that population? In our T1, our next generation, we're going to have exactly 1,000 out of 1,000 total alleles, okay? So here's the question for you. How do we determine the probability of transmitting from one generation to the next, from generation 0, transmitting 500 of 1,000 alleles? So we have 1,000 total alleles that are going to be transmitted, and we want to ask, what is the probability that 500 of them are the big A allele? So we could think about this as each of these transmission events is perhaps a trial. And with each of these transmission events, the transmission of a big A allele would be a success. So if I'm asking the question, out of 1,000 trials, I have some probability of drawing a big A allele, and I want to ask, what's the probability that 500 of those alleles will be the big A allele? How can I compute that? If we go back into the recesses of our mind, maybe just from a few weeks ago, These are Bernoulli trials. We're randomly drawing from our population, but we can use the binomial probability to be able to ask, what is the probability of randomly drawing 500 big A alleles from 1,000 total alleles is going to be 1,000 choose 500. Did I write that correctly? Multiplied by our probability of a success, which is 0.5, to the power of 500, multiplied by our probability of a failure, which is 1 minus 0.5 in this case, to the probability or the number of failures. And I can compute that probability is going to be equal to 0.025. So here the question I've asked is, what is the probability that the frequency of the A allele stays the same in our next generation? And I've used the binomial probability to compute that. And I see that, in fact, the probability is very low that it's going to stay the same in the next generation. Through this process of random sampling, the allele frequency is going to change in some way, i.e., it is pretty unlikely that P will stay the same. And so we can use this same concept now to simulate drift across time, across multiple generations. I'll do us a favor here. I'm not going to consider such a large population. Let's imagine 1/10 the size. So let's imagine n equals 10, a population size of 10. And at our t0, we can ask, what's the probability of drawing 5 alleles? So the probability that 5 of 10 big A alleles are drawn. The way we would compute that is 10 choose 5, 0.5 to the power of 5, and then 1 minus 0.5 to the power of 5. And to do that, I could look here at the probability now of either selecting 0, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. And if we look at the binomial distribution, we expect to see a distribution that looks like this, with highest probability at 0.5, but all these other possibilities of either drawing 0 or 10 are also possible outcomes, okay, with probabilities that we can compute based on the binomial distribution. So the probability of pulling 5 big A alleles out of 10 is going to be 0.246. And so actually it's more likely that we're going to randomly draw some other number of big A alleles from this population. Okay, it's more because here's our probability, our maximal probability of drawing 5, but we could either draw 0, 1, 2, 3, 4, or 6, 7, 8, 9, 10. And the probability of drawing one of those numbers of alleles is going to be 1 minus 0.246. And so we can use this to be able to now say, okay, well, we could randomly draw from this distribution. And in fact, in our next generation, we randomly draw 2 alleles. And if we do that, then now with some probability that we can compute, then we now have, we still have n equals 10, but now we have 2 copies of A, and so P equals 0.2 in this case, suddenly in our next generation. And so now we can ask the same question, well, when we form our next generation, what's the probability that the allele frequency is going to stay the same in that case? Well, now we can say, in that case, what's the probability of drawing 2 of 10 individuals? We need to use the binomial again, but now our P is equal to 0.2 to the power of 2, 0.8 to the power of 8. And now we can look, if we draw our binomial distribution between 0, 1, 2, all the way to 10, but now with probability 0.2, it's going to have some maximal value at 0.2, but all these other possibilities can also occur, and we can compute this probability, but we can also randomly draw from this probability distribution and see it's possible that we would get now 6 in the next generation. And so we can continue this process of binomial sampling to be able to look at how allele frequencies can fluctuate across time, just as simply as a function of random sampling from the population. And so through this process, we call it genetic drift because when we look across time, we see the allele frequencies going up and down. They're drifting up and down, just like a daffodil, drifting through the air. But ultimately, through this process, we can easily lose alleles. That is when we randomly sample and we get a 0, or they can go to fixation. And that in this scenario would be when we randomly chose 10 with some probability that we can compute. And this is going to depend, the probability this will depend on the population size, N, okay? And so what we're going to do in our computing exercises is explore this by looking at how we can use computer simulations of genetic drift in populations of different sizes to see how population size impacts these dynamics across time. And what we're going to see and try and see empirically in our simulations is the answer that I'm going to give you right now is that the probability of an allele going to fixation ever, not in one generation, which was the first example I showed you, but the probability that ultimately it will go to fixation, the probability of fixation of an allele is equal to its frequency. And this is a principle of genetic drift that we'll observe empirically using simulations. Okay, and so what this literally means is that if we have an allele at 0.1, then the probability that it will ultimately go to fixation is equal to 0.1. And so if we think about populations in general, and we think about the introduction of new variation through the process of mutation, so if we introduce a new mutation into a population, what is the frequency of a new mutation in a population, in a diploid population? That frequency is going to be equal to 1 over 2n, the size of the population, okay? So if we have n equals 10,000, that means that our frequency is 1 in 20,000, which is equal to 5 times 10 to the negative 5, which is equal to the probability of it ever going to fixation of a new allele. So it's a very low probability, but it is non-zero. And we can ask the converse, we can ask for a new allele, what's the probability that will be ultimately lost from the population, is just going to be equal to 1 minus the probability that it will be fixed, so 1 over 2n, which equals 0.99995, okay? So highly likely that it will be lost from the population, a new mutation will be lost from the population, but not guaranteed. There is some small probability that ultimately it will become fixed in the population. And this is going to be important when we start thinking about long evolutionary times and sequence divergence between humans and closely related species, and looking at fixed differences between those two species. And so the consequences of genetic drift are that slightly deleterious mutations, so less than ideal variation, slightly deleterious mutations, can be brought to fixation just through this process of random sampling. And similarly, advantageous alleles, so if this mutation was beneficial, the new mutation that's introduced into the population, it can also, advantageous alleles can be lost through drift. All right, so this is one of the key factors that controls allele frequencies across time, is just this process of random genetic drift, okay? And we're going to study how the strength of genetic drift differs as a function of the population size. And what we'll see is that, again, the same principle of the law of large numbers is at play here, which is that as the size of the population increases, the probability that if we're at allele frequency 0.5, that we'll get exactly allele frequency of 0.5 in the next generation is less, because there are so many other possible outcomes. However, the variance in the allele frequency in the next generation is going to be reduced as the sample size or the population size increases. And so we'll study the consequences of this principle for allele frequencies in populations of different sizes across time. All right, so now I want to talk about the second force that acts on the fate of alleles and populations. And so this is the non-random force of selection, Darwinian selection. So this is the process, of course, by which individuals with certain heritable features are more likely to survive and reproduce than other individuals that lack those features. And so this force of selection impacts the frequencies of alleles in populations. Okay, and so we talk about Darwinian fitness, which refers to the ability to both survive, but that's not enough. You need to survive and reproduce. Okay, so reproduction is the key metric when we're talking about the fitness of an individual. So you can, I have 2 kids, so my absolute fitness, W, big capital W is a symbol that we often use, is 2, literally. Okay, so the number of offspring that we have. And often we convert this to a relative fitness, which is the fitness relative to all the other individuals in the population. Okay, so if we considered my fitness of having 2 kids relative to the maximal fitness in our population, which let's say was some family with 10 kids, then my fitness would be 0.2 relative to the maximal fitness. And so let's talk about the Darwinian selection we call a type of directional selection, whereas drift is random and the allele frequencies are going in random directions every generation. Darwinian fitness is directional selection. That's going to move the frequency of an allele in one direction, either until it's completely fixed in the population or until it's lost in the population. And there are 3 ways in which directional selection can act. We can have positive selection. This is what we're often you're familiar with this concept, which is going to bring a favorable mutation to higher frequency in the population. So in the case of complete dominance at the A locus, both the heterozygote, big A, little a, and the homozygote, big A, are more fit than the little a individual. And so that's going to increase the frequency of the big A allele in the population. And this can occur either in this scenario of complete dominance or even in the scenario of where the heterozygote is intermediate, so it has a fitness less than the big A allele, but it's still more fit than the little a homozygote in this case. And in this case, this is going to drive the frequency of the big A allele to increased frequency in the population. The most pervasive source of selection actually is not positive selection driving, advantageous alleles, but is a phenomenon known as purifying selection, right? And so this is selection that acts to conserve the function of genes and gene sequences that are beneficial. And so this is selection that's removing deleterious alleles from the population. So most new mutations are actually going to be maladapted or deleterious. And so purifying selection acts to remove these alleles from the population. And so this is what prevents adaptive traits from being degraded, right? So if you have a mutation that reduces the some trait that's beneficial that exists in the population, there's going to be selection against that particular individual. And this was, Darwin referred to this as the rejection of injurious variations. He didn't know anything about the molecular basis, but he theorized, an important part of his theory was that selection was acting to remove these individuals from the population. And then the third type of directional selection is a really interesting phenomenon known as balancing selection. And so this is selection that maintains polymorphism in the population. And so this is in the phenomenon, one example of this is the phenomenon in which the heterozygous class has higher fitness than either of the homozygous classes. And so natural selection is going to favor the maintenance of both alleles because it's going to favor the propagation of heterozygous individuals. And this is a really interesting phenomenon that's going to move the allele frequencies to some equilibrium point in the population in which both the alleles are maintained. And so we can visualize this as sort of a directional selection, but it's selecting for maintenance of diversity. So moving along now, I want to consider how selection is going to act to change allele frequencies across time. So we can, let's look at that scenario here. Okay, and so we can expand our understanding of Hardy-Weinberg equilibrium to think about how selection on top of the principles of Hardy-Weinberg equilibrium are going to alter allele frequencies across time. And so let's imagine here in our first case, case study one, we can imagine a population. So we have a population with P equals 0.1 and Q equals 0.9. That is in the Weinberg equilibrium, okay? But in this first case, we want to consider the scenario in which the A allele is the favored allele and it is dominant, okay? And so what that means is that the AA genotype and the big A, little a genotype are equally fit, and they're twice as fit as the homozygote for the recessive allele, okay? And so this is, they have equal fitness, and there is selection against the homozygous recessive allele. And again, we're going to think about the implications of this differential fitness across generations as each new generation is being formed. And so to do that, let's make a table. Let's think about our generation at T0. And let's think about our three different genotypes. So we have big A, big A, big A, little a, and little a, little a. And I told you that the fitness of these two genotypes, big A, and big A, little a, are twice as fit. And so we would summarize that as the average absolute fitness, or the number of offspring, which is going to be our capital W. So these individuals on average have 4, these have 4, but these individuals only have 2 offspring. But we want to convert that to a relative fitness, our lowercase w. And as I told you, that's relative to the maximal fitness. So these individuals have maximal fitness of 1, and this individual is 0.5. All right, I told you what the allele frequencies are, 0.1 and 0.9, and that the population is in Hardy-Weinberg equilibrium. So who can tell me what the genotype frequency is of the big A allele, genotype, or the homozygote, in the chat? Big A. We're in Hardy-Weinberg equilibrium, and you all know how to compute genotype frequencies given the allele frequency based on Hardy-Weinberg equilibrium. Has anyone got a frequency for me? 0.1 squared, which is what? 0.1 squared is P squared, right? We get our P squared. That's 0.01 in this case. And similarly, our little a genotype frequency, what's that going to be? 0.81. Okay, good. We got our 0.81 is our genotype frequency. And of course, our heterozygote, how do we compute that? 2pq, is that right? So 2pq, p times q is, so it's going to be 0.18. Is that right? Have I done that correctly? Yes. And what should those genotype frequencies sum to? 1, right? Okay, everything's good. So those are our genotype frequencies in the population that's in Hardy-Weinberg equilibrium. But now we need to compute the contribution of those genotypes to the next generation. So we can think about the relative contribution to the next generation. And to do that, we calculate our fitness multiplied by the genotype frequency, because these individuals are going to contribute a relative frequency of 1, whereas these individuals they're going to contribute half as many individuals to the next generation. So that's 0.5 times 0.81. And these individuals, again, are going to be 1 times 0.18. So we do those calculations, 0.01, 0.18, but this, in this case, is 0.405, half as many offspring from these individuals. Now, if we add up these three numbers, we sum them to 0.595. And so what we need to do is convert that so that they sum to a frequency of 1, okay? And so we can talk about the effective genotype frequencies. And to do that, we just normalize by dividing by this number. So we take 0.01 divided by 0.4595, and that equals 0.017. We take 0.18 divided by 0.595, that equals 0.302, and we take 0.405 divided by 0.595, and that equals 0.681. And now if we sum these effective genotype frequencies, they sum to 1. So remember, with Hardy-Weinberg equilibrium, we can compute the genotypes based on our expected genotypes in Hardy-Weinberg equilibrium based on the allele frequencies. And if we have the genotypes, we can compute the expected allele frequencies. So in this case, we can now say, we can calculate the expected frequency of alleles in the next generation on the basis of the effective genotype frequencies that's accounting for this differential fitness. And so how do we do that? Well, we can say in the next generation, the allele frequency of P, which I'll use P1 for our first generation following the 0th generation, that's going to be, remember that we take all the individuals in the homozygotes, so the allele frequency of 0.17 plus half the individuals in the heterozygotes, 0.302, which equals 0.168. And now if I compute the Q, which I could easily do by 1 minus 0.168, but let's just check that it's this number, all of these individuals, 0.681, plus half the heterozygotes, do the math, 0.832, sum them up to make sure that the allele frequency is equal to 1. Okay.
I have a question.
Yeah.
Is it 0.017?
It is 0.017. Sorry about that. Thank you. Which means, but this allele frequency is still correct, right? 0.168. Yep. Because we started in this generation, P0 and Q0, with an allele frequency of 0.1. But now, because of this differential fitness and our calculation of the effective genotype frequencies here, we can compute what our allele frequencies are going to be in the next generation. And that's increased gone from 0.1 to 0.168, right? So we can ask, what is the change? Therefore, what is the change in P in just one generation because of this differential fitness? And we can just say, well, that's P1 minus P0, which equals 0.168 minus 0.1, which equals 0.068. And so the allele frequency is increased by 0.068 in that one generation. Or if we wanted to talk about that as a fraction of the initial frequency, it would be 0.068 divided by 0.1, which equals a 68% increase in allele frequency in one generation due to natural selection. And so we can continue this calculation now. We can ask by just repeating this process by asking now what happens in the next generation. So we're going, as we go from generation 1 to generation 2. Okay, so now let's think about, now we're in generation T1. And again, we can make our table where we talk now, we can forget about the absolute fitness. Let's just talk about relative fitness. W of our genotypes, big A, big A, big A, little a, little a, little a. And we have this scenario in which the selection against the homozygote recessive. We can compute now our genotype frequencies given our allele frequencies, which are up here of 0.168 and 0.832. So we know then to compute the big A genotype frequency, that's going to be 0.168 squared, which equals 0.028. This is going to be 2 times 0.168 multiplied by 0.832, which equals 0.279. And this is going to be 0.832 squared, which is going to be 0.692. And those are going to sum to 1, as we expect for Hardy-Weinberg equilibrium. But now again, we want to talk about the relative contribution to our next generation. So the relative contribution, we're making the calculation based on the fact that these two genotypes are going to contribute twice as many individuals to the next generation. And so we're going to have, we can do this calculation this way, 0.028, 1 times 0.279 equals 0.279, but only half of this genotype are going to contribute. We're going to contribute half as much to the next generation, which equals 0.346. If we add up these three numbers, the sum equals 0.653. And so we need to normalize again to get our effective genotype frequencies by dividing 0.028 by 0.653 and getting 0.043, 0.279 divided by 0.653 equals 0.427, and 0.346 divided by 0.653 equals 0.53. And if we add these three together, then we have our frequency sums to 1. And so now, again, we can go through this process of computing the expected allele frequencies in generation 2. So P2 in this case is going to be all of these individuals, 0.043, plus half of our heterozygotes, 0.427, that equals 0.2565. And Q2, Q in our next generation is going to be 0.53, which is this number, plus half of our heterozygotes, which equals 0.7435. And our allele frequencies sum to 1 as they should. And again, now we can ask, what is the change in P between generation T1 and T2? Now P2 minus P1 equals 0.2565 minus our frequency in generation 1, which was 0.168, and that equals 0.0885 in this case. So in this scenario, it changed by 0.068 in one generation, and now it's changed, the allele frequency has changed by 0.0885. And so what we can do and what we will do is now think about time on our x-axis and P, our allele frequency, on the y-axis, and we can see that we started at an allele frequency of 0.1 in the 0 generation, and then we went in the first generation, we went up, and in the second generation, we went up. And who predicts what's going to happen in the third, fourth, fifth, and sixth generation? It's going to keep going up, right? With some interesting dynamics until we get to what point? When's it going to stop going up? When we get to an allele frequency of 1, right? So it's going to go with time until we get to an allele frequency of 1. And we're going to watch those dynamics for this particular scenario. And we can do that by hand using exactly this algorithm that I've displayed here and been through 2 iterations of, or we can do this in the computer and write a function in R to be able to simulate this across time. And that's exactly what we will do. All right, and we're going to contrast that with interesting cases. So for example, in case study 2, we could ask the question, what if the recessive homozygote is the one that is favored? And again, if we had P equals, let's say P equals 0.9 and Q equals 0.1 in this case, and we talk about the relative fitness of the big A allele, the big A little a genotype, and the little a, and in this case, if these individuals have half the relative fitness of this individual, we can again compute the expected genotype frequencies. So let's just see what that would be. The genotype frequencies in this case, P squared 0.81, Q squared 0.01, and 2PQ 0.18. We can compute the contribution to the gene pool, which is going to be half as much for these individuals, half as much for these individuals, and a relative contribution of 1 for these individuals. We compute our effective genotype frequencies, which I'll tell you are going to be 0.802, 0.178, 0.0198. And then we can ask what is the frequency in the next generation, which is going to be 0.802 plus half of 0.178, which equals 0.891, whereas Q is going to be 0.0198 plus half of 0.178, which equals 0.1088. And so the change in Q in this example is Q1 minus Q0, which equals 0.1088 minus 0.1, which is 0.0088. And so we have a much smaller increase. And what we will see is that it looks like this will be much slower to go to fixation. And we can study those dynamics in this scenario. And we can do that in the computer. Our third case, which we're going to leave for the computer, but is the most interesting case, is what if selection acts on the heterozygote? And this is our case of balancing selection, in which we have the relative fitness of our genotypes is as follows. And we can use exactly the same procedure to be able to study what's going to happen to these allele frequencies. And I think something very interesting will happen in this particular scenario. All right. And so take-home message is that selection can act on alleles to drive them to alter their allele frequencies in a directional selection manner. And we're going to study how that the dynamics with which that's going to change allele frequencies across time, given different scenarios. And this has very real-world applications for understanding allele frequencies in human populations. So a classic example is the example of sickle-cell anemia, in which the heterozygotes have a fitness advantage because heterozygosity for the sickle cell trait confers, or the sickle cell allele confers a resistance to malaria, whereas homozygosity for the sickle cell allele results in being afflicted with this condition of sickle cell anemia. And so this is a real world example of balancing selection, where selection is acting to maintain the polymorphism in the population, despite the fact that homozygotes for that polymorphism have a fitness disadvantage. And so what we've covered today are the two main forces that are acting on allele frequencies in populations. So drift, random genetic drift, which causes allele frequencies to go to loss or fixation, depending and as a function of population size. And then directional selection, which can act either to drive allele frequencies to fixation or to loss, and interesting scenarios in which we can have selection either for maintenance of polymorphism or the converse scenario in which we have selection against heterozygotes. And that would be another really interesting scenario, which we're not going to study in our simulations, but would be an interesting case to look at once you're able to do the scenarios that we'll look at. And of course, underlying this is the assumption that new mutations are being introduced into a population at some rate, and this is due to errors in DNA synthesis. So we get new variation introduced as a function of mutations, which can also remove variation from the population. And another source of new variation in population is when through migration. So when new individuals move into a population, they can bring in new alleles as well. And so there are all these different interesting forces that are acting on allele frequencies in populations. And in our assignment, we'll study both genetic drift and selection and how that alters allele frequencies. All right, so there was a lot of material that we went through today, but summation is two distinct forces, drift and selection, that are best studied through computer simulation. And what I've given you is the foundation for being able to do those simulations, to be able to study how that's going to impact allele frequencies across time. So this is expanding on your basis of understanding of allele frequencies in populations based on Hardy-Weinberg's principles.